video, we're going to talk about how hormones can activate G proteins, which can then use calcium as a secondary messenger to still achieve cytoplasmic effects. So let's go ahead and get started. So here we have the extracellular side. Here we have the intracellular side. Here we have a free-floating hormone, and this again can be anything from peptide hormones, catecholamines, and then eicosanoids. Here we have a G protein receptor. Here we have an inactivated G protein. Here we have a calcium ion channel. And here we have our phospholipid bilayer, as you can see. Okay, so let's see what happens when this hormone interacts with this G protein receptor. So as the hormone travels along, it meets the G protein receptor and it binds. Now remember that before it binds to the receptor, the G protein is inactivated. Once it binds to the receptor, the intracellular G protein becomes activated. Now, once the G protein is activated, it is able to activate an enzyme called phospholipase C, also known as PLC. Now, PLC causes the production of another enzyme called diacylglycerol, also known as DAG, and it also creates inositol triphosphate, or IP3. Now, the important thing to know about the IP3 and the DAG is that these are both formed from the phospholipid membrane. So PLC will actually take these and create them from the membrane. So once that happens, the IP3 is able to diffuse into the cytoplasm, and it releases the stored calcium ions within the smooth endoplasmic reticulum or the endoplasmic reticulum, depending on the cell. So now once it does that, the now released calcium ions are able to interact with the DAG. So let me just draw this structure out here. So here we have our smooth endoplasmic reticulum, and the IP3 causes the release of the calcium ions. The calcium ions are then able to travel and bind the DAG. So they interact with the DAG. Now, once it does that, the DAG and because it's now bound to the calcium, produces another enzyme called protein kinase C, also known as PKC. So these two they activate PKC. Now, PKC is also a membrane protein, but once protein kinase C is activated, protein kinase C is actually able to phosphorylate the calcium ion channel proteins. So PKC then in turn travels up to the channel and phosphorylates it. Now remember that phosphorylation is the transfer of a PO4. It transfers it to the channel protein and it opens the calcium channel. This causes cytosolic calcium to enter into the cell. Now, once that enters, the calcium is able to bind to calmodulin. Now, before we get there, one interesting thing to note here is that because the PKC, or the phosphokinase C enzyme, phosphorylates the calcium ion channel and calcium flows in, we'll remember a couple steps back when the IP3 released the calcium stores from the smooth endoplasmic reticulum. The calcium traveled to the DAG and it bound to the DAG, which activated phosphokinase C. So if you look, the more calcium that enters, the more it's able to bind to the DAG. So this sets up a positive feedback system. Now, in other words, the more calcium that there is, the more that it can bind to the DAG, and it will cause more calcium to keep coming in. So in other words, it'll do it until it exacerbates it. So a positive feedback system. Now, once the cytosolic calcium fluxes in, the now abundant calcium binds to an intracellular protein called calmodulin. And calmodulin is then able to activate cytoplasmic enzymes, which then will cause the effects within the cell. Now, these are normally stimulatory effects, and they usually occur with alpha-1 receptors that are stimulated by epinephrine and norepinephrine. So, alpha-1 receptors with effects on norepinephrine and epinephrine. So again, just to kind of recap what happened here. The outside of the cell hormone travels and interacts and binds with the G protein receptor in the membrane. What was an active G protein becomes activated by this binding, and the activated G protein activates PLC. PLC then activates IP3 and DAG, which were lipid membrane bound proteins, or enzymes rather. Once those are created, the IP3 travels to the smooth endoplasmic reticulum and releases the intracellular calcium stores. That intracellular calcium store release then travels and interacts with the DAG, and together the calcium and DAG activate PKC. PKC then phosphorylates the calcium ion channel, and it allows for extracellular calcium to flux into the cell, increasing the calcium concentration within the cell even more. Once that occurs, the calcium then binds with calmodulin, and then that carries out the effects within the cell, or it rather activates cytoplasmic enzymes that then carry out the effects within the cell.